Hello, everyone, and great to be able to talk to you about Triumvira Immunologics, an exciting engineered T-cell therapy company. I want to thank you, organizer, for inviting me to share with you the our technology, the biology, and some exciting data and where we're going from here. As you all know, the advent of engineered T-cell therapy has changed the treatment of certain cancers, forever, we hope. Obviously, we see phenomenal results in heme malignancies like lymphoma, leukemia, and myeloma. However, there are some challenges that have arisen in the space. So if you look at the top right, obviously we know that manufacturing T cells is still very time, dollar, and technology intensive. The risk of severe toxicity still exists despite the use of steroids or anti-IL-6 therapies. And it also means at the end of the day, also sometimes because of the age of the patients, it means that only a relatively small number of patients are eligible right now to receive cell therapy. Reimbursement is an issue because we know they're very expensive therapies. And also, there are so many companies pursuing the same targets, has become a really crowded space if you think about CD19 as an example. But the challenge, and that's why we don't want to talk about is the top left, is the little success so far in solid tumors. And that has to do with the fact that engineered T cell often show tonic signaling, therefore have premature exhaustion, lack of T cell persistence, difficulty going over a hostile tumor microenvironment. And that's what people have come up with complex new designs of, for instance, of CARs for solid tumors, but it doesn't make it easier sometimes for patients who have to tolerate these therapies. So for us, it's all about differentiation. We truly think that a more natural activation of the T cell represents the next step forward in T cell therapy. So Triumvira, our technology is known as TAC, or T cell antigen coupler. And the value proposition is that we expect to show efficacy and safety in solid tumors, which we have proven now consistently across different mouse models of cancer. And we consistently show that the TAC T cells outperform the CAR T cells in those settings, and not only in our own experiments, also been externally validated now by other pharma and biotech companies that we're working with. We intend to bring several product candidates into the clinic in the next 2 years. The first one will be an autologous TAC directed against HER2 for solid tumors, and then also later on an allogeneic TAC for solid tumors. We have not disclosed the targets yet, and we hope to bring that to an IND by the end of next year. We have built a team that has strong engineered T cell drug development and financing expertise, and we just completed a $55 million Series A round of financing. So let's talk about the TAC. The TAC is a chimeric antigen receptor, but structure is quite different than anything else out there. We have 3 domains, a tripartite structure, and because of that, it's really unique and innovative, and that's why we receive very strong compositional meta claims in the US and in other countries. So starting at the top, the most outside presenting domain is a ligand binding domain that's very similar to what CAR. It's either a single chain antibody, it could be a designed anchor repeat protein or DARPin, or any other peptide will work. That domain is linked to a flexible linker to what we call the heart of the TAC, which is a proprietary single chain antibody to the CD3 epsilon domain of the normal TCR. And thirdly, and that's really unique to us, we have the transmembrane and cytoplasmic domain of the CD4 co-receptor. So how does it work on biology-wise? So on the top left, you see a normal T cell receptor. As you know, it consists of a number of extracellular, transmembrane, and intracellular proteins. And on the left hand side of that, you see in blue is a co-receptor, could either be CD4, CD8. That also brings on the inside a little LCK kinase that is important for phosphorylating the various 10 items in the inside of the TCR. Next to it is a CAR. A CAR is a beautiful, simple design, very effective as we know. But the CAR is either on or very on. There's no off switch to a CAR because it has its own activation domain, it has its own co-stimulatory domain. So that basically CAR T cell is always active, and you can see the intracellular side only represents 3 of the 10 items, and that's quite different from the normal TCR. On the right-hand side, you see the normal TCR again on the left, but now you see the TAC T cell on the right, where you see that the TAC, of course, it binds to a cancer-presenting antigen, and then, of course, it binds to the single-chain antigen and brings the normal TCR into play. And then on the inside, you see that the intracellular part of the TAC T cell is exactly the same as a normal T cell. So the LCK kinase helps phosphorylating the 10 items, which is important not only to activate the T cell when it meets up with an antigen, but also to stop the T cell from being activated when it does not need to be activated. On the bottom graph, two of our competitors, that's TCR squared and Eureka, they both also using the normal TCR to activate the T cell. However, we're the only ones that have that CD4 co-receptor domain that helps with the normal activation of the T cell. So what does it mean? So these characteristics that we have seen truly present advantages to the TAC T cell. Obviously, just like a car, it works independently of the MHC complex. That's what you want. However, we don't show any tonic signaling, so we don't see upregulation of, exhaustion markers like PD-1, LAG-3, or TIM-3. Remember, the TAC does not have an activation domain. It does not have a co-stimulatory domain. All the activity goes to the normal TCR. It also, we have shown now through photography that the TAC T cell forms a normal immune synapse with a cancer target cell. We don't see an overexuberant cytokine release, no premature T cell exhaustion. We show very strong T cell persistence over time, deep penetration and activation in solid tumors, and very strong in vitro and in vivo efficacy in tumor models of cancer. And no safety signals to date. And these TAC T cells truly retain a memory T cell phenotype that we truly think is important for adaptive cell therapy. So obviously we've done a lot of work in different tumor models, so I'll share that, mostly focusing on solid tumors in this case. So this first one is an ovarian cancer, OVCAR3 HER2 overexpressing cancer cell line. You inject it in the flanks of the mice, you let them grow. On the left, you see the growth curve. On the right-hand side, you see the body weight curve. And you see on the left, you see that the green, of course, is the controls. They see exponential growth of the tumor. And the CAR, in this case, is the CD28 second generation CAR. You see that the tumor stops growing. You don't see much regression. A 4-1BB, by the way, in this model is not active at all. But with the TAC in the blue line, you see consistency. We see full and immediate regression of the tumor. On the right-hand side, we know that the CAR-Ts are extremely toxic, and you see that in the body weight change of these animals. So they lose almost 25% of their body weight. We have a mouse ICU to try to coach them back to life to be able to finish the experiments time-wise. But you see that the TAC T cell treated animals have no change compared to the control. So there is no toxicity observed in TAC T cell treated mice. So people say, well, how is that possible? So here what we did is we did a multiparametric immunohistochemistry on tissues taken from tumor or a normal lung. You see the same in normal cardiac tissue of these mice. So these pictures, you see TAC, CAR-T, and control cells. And you look at the TAC T cells on the left top picture, you see that the TAC T cells are actively penetrated into the tumor. The proliferating, so the Ki-67 positive, you see both the yellow and cyan, so you both CD4s and CD8 cells, and this leads, of course, to the regression of the tumor. The CAR-T cells, however, 7 days after injection of the cells, there are no CAR-T cells in the tumor. They're distracted by antigen presenting in the lung and in cardiac tissue. And you can see mostly are yellow, so they're CD4s, and we know that CD4s are associated with toxicities. So the CAR-T cells are so trigger happy, they find another antigen, they bind to it, and they start producing cytokines. And they don't really have to challenge the chance, basically, to penetrate into the solid tumor. So a lot of people say, why do TAC T cells penetrate? The question is more, why are CAR-T cells not penetrating? And that is because of the tonic signaling, the trigger happy, and they really attach to another antigen in another place. This slide is the slide taken from the same cancer model. This is the slide that the clinicians like. You see here the production of cytokines between the CAR-T cells in red, the TAC T cells in blue, and then the controls in green. And you see here, remember this is a log scale, that the CAR-T cells produce 100 to 1,000-fold more cytokines than the TAC T cells do. However, the TAC T cells are effective in this model, not the CAR-T cells. So the CAR-T cells overproduce way too much cytokines that you need, and that TAC T cells produce enough interferon gamma and TNF alpha to still penetrate and kill the tumor cells. So this is a summary. We did a lot of work on the HER2 models, whether that's gastroesophageal, gastric cancer. We also looked at a breast cancer model that was Herceptin resistant, and you see phenomenal results repeatedly, and either complete regression. And on the right-hand side, the curve, that's a nice dose response curve, where you see even at a 1.5 million dose levels of cells, you see that there's efficacy in these models. So whether we take 12, 6, 3, or 1.5 million TAC T cells, you see that there's efficacy in this OE19 gastroesophageal cancer model. So where are we now? Obviously, we have strong data in different models. Of course, we keep talking to KOLs about our program. We received FDA feedback on our pre-IND submission. We are basically expecting to submit an IND to the agency in Q1 of next year. So that brings us then to our pipeline here. As I said, the autologous HER2 goes in the clinic early next year, focus on the HER2 positive solid tumors, so ovarian, breast, gastric, and other HER2 overexpressing tumors. We have an allogeneic tag that is built on gamma delta cells because, as you know, we cannot knock out the TCR because we need the TCR for the tag to function. So we're focusing on gamma delta cells and also on invariable NKT cells. We have not disclosed the targets. We have quite an extensive bioinformatics program to looking at different tumor targets and obviously binders for those in solid tumor settings. And we hope to bring an allogeneic tag into an IND by the end of next year. As I said, we're working on quite a few other, allogeneic solid tumor programs. We do have an autologous CD19 tag, very efficacious, very safe. However, when we were ready to start the study, COVID-19 happened, so we didn't start the study, as you can imagine. And right now, we truly are decided to focus on the solid tumor setting. So there's an opportunity if people are interested in licensing our tag CD19 program. So obviously, significant near-term value inflection point for investors and also for growing and expanding the company. Obviously, the focus will be on the HER2 program, followed then by an allogeneic tag program. And as I said, we can definitely run the CD19 study. However, we've decided to postpone that and focus on solid tumors squarely. When the paper on the technology was published in Nature Communications in 2018, the day after the phone started to ring. So all the well-known pharma and biotech companies in the CAR-T space called and wanted to talk. So not only do they want to talk, they also want to put their resources to work. So right now we're doing 3 evaluation programs, one with the top 5 major global US pharma company, and 2 with 2 of the top 3 US CAR-T companies. And the nice thing for us is that in their own experiments, their own labs, they also have shown that the TAC T cells outperform the CAR T cells. So there's a nice external validation. And we're really looking forward to work closely with some of them, perhaps get to a licensing discussion. And on the bottom, it's important. We all know that manufacturing is a big challenge, as I said in the beginning. So for autologous, especially. So we did a deal with Lonza that we announced several months ago, where we have a risk-sharing manufacturing agreement to the use of their Cocoon system, which is a beautiful, ultimately point-of-care manufacturing system of TAC T cells. Beautiful cells that have produced so far, and we're looking forward to bring that to the clinic and use that in our HER2 phase 1/2 study already. So our financing history, the company was founded in Hamilton, Ontario, McMaster University. Jonathan Bramson is a professor of immunology there, together with Brian Bloom, the CEO of Bloom Burton, investment advisory firm. They founded Triumvira in 2015. I came aboard in January 2018. And since then, we've been on a financing route, and we just closed a $55 million Series A that was led by Leaps, Bear, and North Pond Ventures, with Bloom Burton, Viva Biotech, Ocean Pine, C3I, and Haywood Security as additional investors. So we're very happy. It's a great, nice round that will bring us into the middle of 2022. So with that, I want to end it, and I want to thank you for your attention. I'm looking forward to any questions you might have. Thank you so much.